मैं नहीं जाऊँगी, मैं ऑनलाइन ही रहेगा। चीप एंड इम्पोर्टेंट लिया है तो वो तो ऑनलाइन ही रहेगा।
Sab directions, traveler sab. Take cotton. Take cotton. Then
नहीं ऑनलाइन नहीं
अच्छा गागल आज क्या
तभी से यूँ क्यों जा मुझे नहीं
करने देने से
ये तो प्रिजर्वेशन में आता है इसके फाइनल आगे
लेबल करो आदिल
anyway, already you are doing in cell culture, I think you know everything how to label it. Like shaving.
आप देखो ना सर उसमें क्या है जो ये वाला है ना उसमें सारा खुल जाता है एक बार में ही तो सारा टेम्परेचर रोटेट है ना
Yes, good. But this is the procedure what we are following there. Then you can use the forceps. Just pull it. Open that one. And now test it. You labeled already.
बड़ा क्या है Can scalp
Maybe take video. Take this video. We'll sing. Just before that one, you can keep it here and directly, if your lab is nearby that one, take it to the lab, then you can start the procedure. If the lab is far away, then you have to wash with alcohol once, right? Take this.
Add TVS
Wash.
alcohol
6 ml, 6 ml.
yes
This open. Add TVS here. Give it the first one.
From how much old sample you were able to get cells?
How much?
Old, no? Maximum period when was that?
Age 23 years.
No, not from time since period of death. Time since death. You were able to get the
Yeah,
cells.
24 hours a day. We
After
got
that,
the
you were not able to.
Yeah. We didn't try that one because sometimes while processing itself the skin will be peeling off, or sometimes the contamination we can see that one.
Peel of skin. Peel of skin.
This depends on the condition of the animal. Even some animals.
If skin starts peeling off of itself, then it's not possible to
It's
get
not
cells.
possible. Even we will not attempt to derive the cells also, right? This is
So on.
TBS. Just I am giving the alcohol wash once. This is only 30 seconds, right? Then I will give PBS wash again.
70% sir
Yes, 70%. Then again PBS.
Serial wash.
Okay.
That's all.
Two washings is option. Just I am keeping here. Your lab is nearby, you can directly
Right.
take this. Then you add PBS there, right? PBS, DMA, what is the medium is available with you, we added that. Piece should be deep in the PBS, right? No, close the weight.
The chop.
if we don't have PBS available then we can
Then it's right.
Normal.
Antibiotic added, huh?
No, antibiotic, always antibiotic. Okay. Then you have to seal it with paraffin and then keep it in Ziploc cover. Then you can send it to the nearest lab.
It's better to transport on cool in
Yeah,
cool
you have to
condition.
keep everything in the cool 4 degrees on the ice. Use the bubble box and you can send it to nearest laboratory or whatever, or maybe even lab. There you can process it. Then this is the how to collect the skin sample. Is for then if you go to the other tissues, then you have to open the body. Other tissues means like any reproductive tract. If it is male, we can cut. If it is female, no, sorry, it is male.
Male.
You told me female. Okay. Here also same procedure. Testicles also we can collect from here, but in rabbit is the testicles will go move inside and outside, right? Then we have to hold like this. In a animal, that means I think it is outside only. You have to clean, disinfect everything like the skin and everything, shaving and everything. It's better to do the same shaving and as well as alcohol. Then you can cut here similar way, but this you can't keep the PBS. You keep it in one 50 ml tube. That tube you put it on the ice. Don't put
Right.
directly this on the any ice. It will so the skin tissue will become the ischemic and it become necrose, right? It's directly the ice affects on the skin, right? So then it is better to everything on the field. If you want to do carry this there, you can do it. It is nearby your lab, the field station or the activity. It is better to do everything is here. Right?
Sir, can you wait for 1 second?
One.
We'll just push this tail here now. Some people.
Yeah, right. Sure, sure. Yeah. But other people, I don't know. What is this?
Let me calculate. कैसे जब कैसे सारे करके लगता है ये दो दे प्रोसेस करते हैं ना
Actually, come here.
फिर वो तो दो दे वो ताता फर्टिलाइजेशन फायर से तो ये सुना नहीं ना पर फायर
Cut down from the base.
मुझे तो इससे
Yes, sir. Yeah. Cut this. Actually, I want to try to take the whole testicle portion, but unfortunately, by this blade nick, this testicular properties came out, right? Even you can carry this one. Even with testicular tissue, similarly, you can disinfect everything, keep it in 50 ml tubes or any Ziploc cover, put it on ice and send it to the nearest laboratory. There we can process. There also, again, we will remove this entire skin. So anyway, it was happened here. Just I am keeping this one. Now this, see the testicular structure, this is the testicular proper, this is the spermatic cord and this is the epididymis, right? Here this is the last portion, right? The epididymis caput. From there only we are isolating the sperm. We will try it whether it is possible. Maybe I think it is adult only. I think we should get some sperm from here, right? Spirit. Sheep, I used to use the retroactive flash. You can using this spermatic cord, I can inject this media to the acid difference. Then you can make it one cold there, and the spermatozoa comes out. But that is the pure spermatozoa. If you are teasing this one, all the epithelial cells will contaminate. The sperm motility will be disrupted. It's any particle is there, the sperm will go and attach to that
Attach,
particular particle. Because sperm has, there is no reverse, it will go always the front side. It will go and accumulate that one around the tissue pieces. So, periodism is difficult to isolate this vas difference. You don't know how the size of the needles to retrograde flesh. So, there also then again we have to just
in case we want to collect only the testicle material, which immediate
use BMEM, PBS, techniques, kinder.
can sperm
PBS.
survive in BME?
You have adopted that spermatogonia.
Because you don't keep any tissue in the alcohol for a long time. So
No.
not more than 30 seconds. Just you dip it and take it out,
Okay.
right? Otherwise it will kill the cells, right? I mean, what is the question? You're asking something.
My question was, if we are collecting the testicular material and without skin,
Yeah.
which BME do we use?
BMEM you can use.
The safest one.
Safe.
What is
No. You have to remove this epidermis.
It has to be like
This is the two ways. If you want to store this testicular tissue, you can remove the epidermis and keep it in DME. If you want to collect both the things, put it in mice, send it to the laboratory. There you can remove the epidermis. Or first you collect the spermatozoa, then you go for further testicular tissue process.
There are seminiferous tubules, seminiferous tubules.
Yeah, seminiferous. You can take the testicular tissue.
What is its use? Has it been standardized?
Yeah, then you can use it for genografting. Genografting.
Okay,
studies.
genografting.
And even you can use the cells for what is this? You can develop the cell line from even testicular tissue also. See, there are
Which cell line?
fibroblastic cells. The cells have different types. Skin derived fibroblastic cells, they have the different characters. And the testicular tissue, they are different. Maybe liver derived cells, organ, specific organs. This is not a specific
What is
organ.
its use in conservation biology? I mean to say.
That is testicular seminiferous tubule. Yes, you can go for genografting only.
Only genografting.
You can derive the cells, the derived cells you can convert into iPS cells, then you go to further. That is the long-term
Long
research. It is not a short-term research one. This is the fat accumulated here. This is the small portion, this is the epidermis. Yes. Capet, we are collecting because the spermatozoa was stored in the capet region, right? So just we squeeze it just like make it some pressure on the take needle. The syringes are there. Just I'm squeezing this one, just I make some holes on the portion. Holes. The sperm is there, automatically comes out. No sperm. This one.
In GBPS? Yes.